So in terms of protein configuration, we said that a lot of work and research has been done to determining how proteins are shaped, but why is that important? Well, let's talk about it. It says a protein-specific conformation is what's going to determine how it functions. So we want our proteins and we want, all of the molecules in our body to be working the way they're supposed to, right? Well, in order for a protein to function, it has to be in a certain shape or a certain configuration. And here they have two examples or two models of protein conformation. So on top, this is just an example of how somebody might describe a protein, and they're utilizing a ribbon model to describe that. On the bottom, they're kind of using something they call a space filling model. So those are there just to kind of give you a visual illustration of how people look at different types of protein. Well, there's four main levels of protein structure you need to be familiar with. And the names of these structures or the names of the protein structures are pretty similar. So we're starting out with the first one, which is called primary structure. All right, and primary structure is pretty straightforward. It's basically just looking at the sequence of amino acids that are found in a polypeptide. And so if you look at this illustration of this molecule over here, it's very linear, it's very kind of one-dimensional, it's flat. So we're just looking at the sequence of all these amino acids that are making up this polypeptide. Now, the second step, as you might have guessed, is called secondary structure. And so this is where we start to get a little bit of shape to our protein that's starting to form. So it says secondary structure is the folding or coiling of the polypeptide into a repeating configuration. And there's two main types of configurations that might happen here. And the first one is called an alpha helix. So in a few weeks, we'll talk about DNA molecules and the fact that they're shaped like a double helix. Well, you can see here that proteins also take on the shape, or they can also take on kind of a helical shape. Well, another conformation they can take on is this kind of, it looks like folded paper to me, and they call that a beta pleated sheet. So these are just some initial shapes that are going to start to form in what they call secondary structure. Now, tertiary structure is where you're going to start to get some three-dimensional shape to your polypeptide or to your protein. And the reason for that is that the amino acids and their different R groups are going to start to interact with one another. And that they're going to cause a lot of coiling and folding of our polypeptide to happen. So on the bottom you see they've kind of highlighted these beta pleated sheets or these alpha helices. And what's happening is all the amino acids and those R groups located on the amino acids are going to start to interact, which you can see in that bottom right-hand picture. So in the tertiary stage of protein structure, we start to get kind of a three-dimensional shape. Now the fourth and final part of protein structure is called quaternary structure. And so this is kind of the big picture. It's the overall protein that's been formed, and it results from the aggregation of two or more polypeptide subunits. So you can see over here, we have kind of our overall protein that has formed. So looking at it in kind of a big picture, we see or we remind ourselves that molecular structure is really important because without that structure, our proteins or the molecules in our body aren't going to be able to function like they're supposed to. So again, we have four stages in protein structure. The first is primary, followed by secondary, followed by tertiary, and then finally we finish it off with quaternary. And from that point, we can start to see our proteins functioning. They might be regulating something, they might be, adding to some sort of structural component in our body, they might help with different types of movement. Again, they can serve as enzymes or catalysts. All sorts of different functions could be going on. But again, they can't happen unless the molecular structure is sequenced the way it's supposed to be. And an example of that is sickle cell disease, which you've probably heard of. But sickle cell disease is a fatal disease that results or occurs because of a single amino acid substitution in the protein hemoglobin. So here's an illustration or kind of a cartoon of how sickle cell disease happens. Well, we said proteins are formed because they have monomeric subunits that we call amino acids. So on the left-hand side of this picture, we have a normal red blood cell. You can see it down there at the bottom. It's just kind of a circular shape. It looks good and healthy. And on top of that picture, that's the sequence of amino acids that are supposed to be, kind of lined up in that hemoglobin protein. So it says we have a first amino acid that they're calling valine, followed by a second amino acid, followed by a third. And the sequence is perfect in such a way that it produces a healthy hemoglobin molecule. Well, what happens in sickle cell disease is that one of those subunits isn't what it's supposed to be. There's a substitution. And that substitution is going to change the entire shape of our protein. And that is what gives us, if you look at the bottom right-hand side of that picture, that kind of sickle cell shape that's going to make those blood cells incapable of taking up the amount of oxygen they need to allow for survival or to allow for proper body functioning. So that's an example of why protein structure is so very important. Now, what determines protein conformation? Well, there's lots of different things that can play a role in that. It can depend on physical kind of conditions that are going on in the protein's environment. It can depend on different types of chemical situations. But this term denaturation is something I really want you to be familiar with. And in denaturation, what happens is a protein is going to unravel or it's going to lose its shape. So it's going to kind of fall back down to that primary structure. And what happens is that when it does that, it can no longer function. And so down at the bottom it says most proteins probably go through several intermediate states on their way to a stable conformation. But if we put our protein in an environment that's hostile, if it's not the right physical or chemical condition, then it's going to cause our protein to denature. So here's an illustration. Okay, on the left-hand side we have a normal protein, and then for some reason it's denatured and it's kind of fallen apart back to our primary structure. And a common example of this is if you guys wake up in the morning and you decide to make some eggs. You make scrambled eggs, so you throw them on the hot plate on your stove. Well, what happens is you notice that once you put the egg in there, it starts to change colors. Okay, it gets thicker. Well, that's because all the protein in that egg is starting to denature because you've put it in a really hot environment. Okay, it's denaturing because it can't withstand those physical conditions you've put it under. Now, this is just kind of a cool little tidbit that I think is interesting, but there are these little, they're not critters, but I like to call them critters, that they call chaperonins. And chaperonins are protein molecules that assist in the proper folding of other proteins. So it's proteins helping other proteins to form. And you see over on the left-hand side, we have this kind of digital image that they've produced. So you have a kind of a cylindrical bottom that's topped off with a cap. And on the right, you can kind of see how they work. But what happens is that cap will come off of that chaperonin body, and a polypeptide and its primary structure will go into the chaperonin. It puts the cap back on, and while it's in there, that protein is able to kind of go through a conformation that is going to give it the shape it needs to function when released back into the body. So once the protein has kind of gone through its transformations and it's reached the final physical structure it needs to be in to function in the body, the chaperonin will release it, and it will then grab another protein and continue the process. So these are like little robots. I just kind of think they're neat.